The overall goal of this low-field fluorine-19 time-domain nuclear magnetic resonance methodology is to determine the average content of fluorinated drugs in formulated drug products. This is a non-destructive technique for drug development and manufacturing in the pharmaceutical industry.
This method can help answer key questions within the pharmaceutics field in the process of formulating drugs in drug products, such as the average content of drugs in tablets and capsules. The main advantage of this technique is that it is non-destructive and faster than conventional methods such as high-performance liquid chromatography when measuring the average content of drugs in drug products.
Weigh the samples for calibration in the appropriate NMR tubes until the marking line, or around 30 mm in height. Also weigh the formulated drug product samples in the appropriate NMR tubes until the marking line, or around 30 mm in height. Next, place the Teflon standard sample inside a 25 or 26 mm NMR tube. Place the NMR tube into the probe inside the magnet. Allow the tube to equilibrate for 5 to 10 minutes before tuning the instrument to F19. Select Auto O1 under the Commands menu in the NMR software. Repeat the measurement at least 3 times and take the last value of the O1 parameter. Navigate to the Commands menu in the NMR software and select Auto P90 to calibrate the 90-degree pulse for F19 with the standard Teflon sample using the standard automated calibration sequence available in the instrument. For a better signal-to-noise